Today, we're diving into a technology that is seriously changing our world. We're talking about tracking pandemics in real time, exploring the hidden DNA in our environment. This is the story of how we're learning to read the code of life faster than we ever thought possible. All right, let's get into it. So, to really get just how big a deal this is, let's start with a situation where every single second could mean life or death. So, picture this. You're right there on the front lines of a brand new outbreak. A dangerous virus is spreading fast, and to stop it, you have to know its genetic code. You need to know what you're fighting. So, what's the plan? Well, until recently, the answer was, honestly, it was slow. Really slow and frustrating. The old way, meant you had to take these precious samples, pack them up super carefully, and ship them off, sometimes across entire countries, to some big centralized lab. A lab filled with these huge million-dollar machines. And the whole thing? It took days, sometimes even weeks. And when you're in the middle of an epidemic, a week is an eternity. It's time you just don't have. But then, everything changed. It was a total game changer. During the West African Ebola outbreak, scientists on the ground pulled off something that was, frankly, revolutionary. They were getting that critical genetic data in less than 24 hours from getting a sample. And the sequencing itself? Get this. It took as little as 15 minutes. That is a monumental leap. And it was all thanks to a lab small enough to fit in their pocket. So what is this thing? This device that basically shrinks a whole state-of-the-art genetics lab into something you can hold in the palm of your hand. I mean, just look at the difference here. On one hand, you've got the old-school sequencers. We're talking room-sized machines stuck in a lab and pretty slow. And on the other hand, you have this new reality, something portable, ready to go anywhere, and spitting out data in real time. It's not just an improvement, it's a completely different way of thinking about science. And here it is. This is the device that's making it all possible. It's called the Minion, and it is wild. It's about the size of a big USB stick. It weighs just 90 grams. That's less than a deck of cards. And you just plug it straight into a laptop for power. It's been out since 2015, and a single one of these can generate a crazy amount of data. I know it sounds like something out of a sci-fi movie, but this is real. It's being used by scientists all over the world, right now. Okay, so we've seen what it does, which is amazing. But how does it do it? How in the world do you read the code of life with something so tiny? Let's break down the science, which is actually super elegant. The secret, believe it or not, is right there in the name, nanopore. A nanopore. We're talking about a teeny, tiny, microscopic hole. It's actually a protein that's stuck in a membrane. And when I say tiny, I mean try to imagine something 100,000 times smaller than a single human hair. It's just big enough for one single strand of DNA to thread its way through. It's mind-boggling. Okay, so here's how the process works, and the core idea is surprisingly simple. First, you've got an electrical current flowing through thousands of these little nanopores. Then, a strand of DNA gets pulled through one of them. Now, here's the magic. As each base of the DNA, the A, G, C, and T, goes through the pore, it blocks the current just a little bit differently. Each letter has its own unique electrical signature. The device just reads those disruptions as they happen in real time, and that's how it spells out the DNA sequence. It's almost like it's reading a strip of Braille, but for the very code of life. So reading those electrical signals is the first step. But turning that raw data into an actual accurate genetic sequence, well, that's a whole other puzzle. And solving it takes some seriously clever computing. See, the raw signal that comes out of the machine isn't a neat list of A's, G's, C's, and T's. It's this messy, wavy, continuous electrical signal. And scientists have a great name for it. They call it a squiggle. So the million-dollar question is, how do you take this noisy squiggle and translate it back into a perfect DNA sequence? The answer is a process called base calling, and this is where artificial intelligence takes the stage. Specifically, they use something called recurrent neural networks, which is a type of AI that's kind of modeled after our own brains. It's fantastic at understanding sequences, things that happen in order, like language. Or, you guessed it, a squiggle. The AI is trained to recognize the unique patterns in the squiggle and translate them into the right DNA letters with amazing accuracy. And what's really cool is that it's not a one-size-fits-all thing. Scientists can actually pick the right AI model for the job. It's like if you're out in the field and you need answers right now to track a virus, you might use the FAST model. But if you're in a lab trying to piece together a whole new genome from scratch, they call that de novo assembly, or hunt for a super rare mutation, you can switch to the super accurate model. We're talking over 99% accuracy. It's all about matching the tool to the task. Okay, let's step back for a second. We've got this tiny, portable, real-time sequencer that's powered by AI. So what? What does that really mean for us, for the world? Well, it means we can now start reading the DNA of pretty much everything. Think about our hospitals. This tech is an absolute game changer in the fight against superbugs. You know, those bacteria that are resistant to antibiotics? Instead of waiting days for lab results, doctors can now figure out exactly which bacteria they're dealing with and what its weaknesses are in less than 6 hours. That means they can give the right antibiotic right away, which can literally save lives. They can change a patient's treatment plan in near real time. And in the world of pure research, it's just as revolutionary. Because nanopore sequencing can read really, really long stretches of DNA, it's helped scientists finally crack the most difficult, repetitive parts of our own genome. This led to the first truly complete map of a human genome. It's also the only technology out there that can read RNA, the messenger molecule for DNA, directly. That's opening up whole new areas of biology we couldn't explore before. Then, you take it out into the wild, and its portability just blows the doors off what's possible. Researchers have these fun-named workflows like What's in My Pot, where they can literally just scoop up some water from a river, run it through the Minion, and see a list of all the living things in there. All the bacteria, viruses, you name it. It's being used to map out the life in rivers across 3 continents. We're getting a snapshot of our planet's biodiversity like never before. And really, all of this, it comes down to the mission of the company that brought this tech to the world, Oxford Nanopore. Their goal is so simple, but at the same time, it's incredibly ambitious. They want to, quote, enable the analysis of any living thing by any person in any environment. And that just leaves us with this one huge, fascinating question, right? We're heading into a future where reading DNA isn't something that's locked away in a high-tech lab anymore. It's going to be in the hands of doctors and clinics, ecologists in the rainforest, maybe even astronauts in space, when anyone can sequence any living thing, anywhere. What are we going to discover next?